Namaste everyone, welcome to our Apoptosis series. This is episode 2. I am Dr. Kalpna Rakholiya and today we are going to discuss about history and discovery of apoptosis. In last lecture we discussed about what is apoptosis, how this word is derived, and today we are going to discuss about the history. So before we start, let's discuss about the apoptosis. We all know every day nearly 50 to 70 billions of cells die inside our body. If cell continuously die, why don't we become thinner every day? Because new cells are continuously being made and it's a balance. So how does the body know which cells should be live and which should be die? So this is the one kind of very precise kind of the death or suicide of the cell, and it occur a very genetically biochemical process occur and cell become a suicide without any harm of the our bodies. So let's start from the early pioneers of the 19th century. In 1842, first description of a natural cell death by Carl Vogt. He just described how apoptosis will be occur, how naturally cell die. Later on in 1885, Walter Fleming, he discovered pioneer of the mitosis, beautifully documented the systematic process of programmed cell death in the tissue and laying the morphological groundwork. Later on, John Kerr, he observed in 1665 in electron microscopy, observed the liver tissue. He noticed dying the cell look completely different from the necrotic cells. So necrotic cell we discuss in our next episode. So it is a completely dying necrotic from the different from the necrotic cell. How? So he observed into the microscope. The first cell are becoming the shrink, then their chromatin become a condense. Afterward, plasma membrane form the blabbing. Then formation of membrane bound small apoptic body, and this apoptic body is cleared by macrophage without any inflammation. So this remarkable observation done by John Kerr. And then apoptosis word was coined in 1972 by three remarkable scientists, Kerr, Weil, and Curie. So, apoptosis is a basic biological phenomenon with the wide range of implication in tissue kinetics. So, he gave the name apoptosis. It derived from the Greek word like leaves falls from the tree, petals fall from the flowers, fruits naturally detach from the tree. Same vessel also die naturally, and this process is now he give the term they give the term apoptosis. So what they published research paper, major contribution of the published in 1972. They demonstrated that apoptosis is an active biological process. It is a differ fundamentally from the necrosis process. It occurs during the normal physiological process, and it is a tightly regulated, and it remove unwanted cell without causing any inflammation. Then after Andrew Ville, it made the another major discovery. He showed that the DNA from apoptotic cell break into fragment. It is approximately 180 to 200 base pairs. So here you can see that this is a DNA structure. This DNA structure cleave into small fragment, and this is the hallmark of apoptosis process. This fragment is between 180 to 200 base pair only during the apoptosis process. So when analyzed by gel electrophoresis, this fragment found the characteristics DNA ladder. So this has become one of the earliest laboratory marker or hallmark of the apoptosis process. Later on, C. elegans revolution occurred in 1980. Scientists turned to the tiny nematode worm. This nematode worm is known as C. elegans. C. elegans body is entirely transparent. This is a soil worm. Every cell lineage is known. Cell death occur with remarkable precision. How? When C. elegans is developed from egg tool in larva sets. At that time, There are 1019 cells are generated, but during the development of that, during development 131 cells are undergo the apoptic process. It means that 131 cells are die by programmed cell death, and only during the adult stage of C. elegans only remain 959 cells, 959 cells remain. So you can see that what about this 131 cell from 1090 cells. So these all cells are undergo apoptosis process, and these cells are not die accidentally or that, but it is a very sequential manner, genetically level, biochemical level. These cells are die. So this made the worm is the ideal model for uncovering the gene controlling apoptosis process. So later on, the three key enzymes were identified from the C. elegans, CED3, CED4, and CED9. CED3 is responsible for this is the one kind of protein is the executor for the apoptosis process. It also found in mammal. Then CED4 is activate the CED3. It function like a apoptic factor 1. CED9 that prevent unnecessary cell death, and it equivalent is like a BCL2 family. So this discovery so that apoptosis is genetically programmed. Later on in BCL2 inhibit the apoptosis process that is discovered by David. It link defective apoptosis to cancer. If apoptosis process is not occur inhibited by BCL2 protein, then cancer it will be arise. Then in 1990, caspase identified multiple group of caspase enzymes are involved. During the no, when apoptosis process is not occur, then this it in a procaspase form. Later on it activate into caspase, and it involve in apoptosis process. In 1990, intrinsic and extrinsic pathway were defined in which multiple proteins are involved. Complete signaling framework is occur, and many scientists are involved too during this pathway, involved in discover this pathway. And later on in 2002, these three scientists, Benner, Horvis, and Sulson, got the Nobel Prize by recognition of apoptosis in a genetical level. So these are the MCQ. You have to solve this question and give me answer in the comment box. Thank you very much for listening this episode.